Thank you all for joining. Just a reminder, chat or raise hand function will work to get you into the conversation or your comment or question into the conversation. But we deeply appreciate your being here today. This particular topic today is rather critical. It is part of a process that we started over a year ago to think through how we can move towards prevention in the Alzheimer's space. Prevention either of symptoms or prevention of pathology, secondary prevention or primary prevention. And that will encompass a number of activities. Initially, we had conversations with about 10 companies and 4 regulators, which produced really an interesting set of thoughts. We have incorporated now Operation Warp Speed and active clinical trial experienced people to think through the whole stream of activities that may be needed to do what we are now calling the Alzheimer's Prevention Accelerator. That's the name of the work group, as we've named it now, to make sure that we encompass all of the kinds of technologies that might be useful, either in, as I say, secondary prevention, prevention of symptoms, or primary prevention, prevention of pathology, however defined as we go through this conversation. That will include not just the conversation we have today, which will be critical, which I'll open in a second, but also target product profiles, optimum clinical trial designs, the breadth, geographic breadth of clinical trials to assure that we have safe and effective prevention technologies that could be applicable globally, and that we begin to think through the challenges of consumer adoption and adherence to these products to get the maximum benefit for patients around the world, 80% of whom will in the coming years be in low and middle income countries. So this is a patient-led effort out of the corporate roundtable of Us Against Alzheimer's, namely the Global CEO Initiative on Alzheimer's. So it is important. It is a part of a stream of activities which appear to be on the agenda with this new administration of moving from treatment of a disease after symptomatic presentation to one in which we try to prevent disease before symptoms arise or before underlying pathology of diseases occur. So we believe in the Alzheimer's field that we will find a receptive voice on that front, potentially with the new administration. But that time will tell on a lot of that. So I would like now to introduce to all of you who all of you know Dennis Selkoe, because one of the topics that did come out of this whole effort to try and move to prevention over treatment is the need to think through surrogate endpoints, the need to really think through how it is that we can, as a matter of science, understand the underlying pathological drivers of this disease in a way that can give regulators confidence that they can approve a prevention product based upon a surrogate endpoint rather than requiring what may be uneconomic and enormous clinical trials over many, many years and maybe decades to assure that the clinical benefit emerges. So that will require both regulatory thoughts about surrogate endpoints, but also potentially post-market requirements that would require follow-up and real-world evidence. But 2 of the targets in the current panoply of prevention technologies are amyloid and tau. And Dennis Selkoe, as most of you know, is one of the deepest thinkers and smartest thinkers in the area of amyloid pathology, its nature, its course, and its essential being inside this disease. And so we asked him to draft a paper on the subject of surrogate using amyloid lowering as a surrogate for clinical benefit to justify traditional approval, final approval. And he, a number of co-authors, I've watched this in draft form over several, and I'm thinking several drafts, with a lot of comments from very smart people in the field, came in and ended up producing a paper with multiple co-authors that does now describe at least the thesis for, and some of the arguments against, and some of the counter to those arguments against in this paper. And so I'm delighted to have Dennis Selkoe with us to start this conversation. This will be an effort to really stimulate the thinking in the field about using amyloid lowering as a surrogate endpoint for traditional approval. And we recognize that it does not happen just because very eminent people in the field of science of Alzheimer's say it. It has to be a dialogue, and this is the start of what will be a really good conversation today. And then we will roll out a number of different ways in which people can comment on this this paper and on the thinking behind the paper as we move forward in the next several months and years. So, Dennis, I've said this before. I'll say it to this wider audience. You have been masterful in drafting a paper and then taking in a lot of incoming comments and then absorbing them as in part of this paper. And you put in a lot of time and a lot of effort. So I not only commend you on your brilliance, but also on your persistence in getting this done. So thank you. And I look forward to hearing how you present this paper and thesis orally today. And then we'll take all sorts of comments and questions from the assembled audience.
Great. Thank you, George. And I'm delighted to begin a discussion of this topic in a formal sense, where my co-authors and I have published this back on November 13th in Alzheimer and Dementia. And you can look at the article. It has lots of references and get the details. I'm also pleased that Jeff Cummings I see on the call, and I suspect there may be other colleagues and co-authors on the call. The goal of this work was to synthesize the views of a number of people. It was not intended to be a sole author publication by any means, and we've actually arranged the authors who contributed directly in alphabetical order, and they all contributed to the improvement of this argument and the thesis of this, and offered a number of suggestions and improvements that we incorporated. So this is a type of a consensus among obviously a relatively small group of experts, a dozen of us. I'm sure there would have been many others who would have joined us had we designed it that way. And so I'm happy to speak for that collective group of authors. We don't agree precisely on every single element of this thesis, but we have come to considerable consensus and modified the document over time, as we also did for our 3 peer reviewers who read this and who were quite thoughtful and diligent in their critique of the original version. The main goal of this paper and this discussion today is to make everyone more and more familiar. I think much of the audience on this webinar already is, with the idea that amyloid accumulation in the brain over time is unequivocally a negative influence on organ function in the brain, and something that needs to be addressed medically as vigorously as possible. Throughout my long time of studying amyloid in the brain, and I'm sure many others would say this also, there's been a huge amount of debate about whether amyloid is causative, whether it is even a key driver of the disease or a minor driver, whether it occurs secondarily or even in a tertiary sense in the disease, not as an early instigator, and whether it is a worthy therapeutic target. And we know that just a few days ago, the European Medicines Agency has come to a decision to allow the use of lecanemab, Leqembi, in people with amyloid buildup who are proven to have Alzheimer's disease by biomarkers. And that, I think, is a great development, and I very much applaud the EMA's decision because it joins a number of other countries that now allow the offering of amyloid-lowering antibodies. We have 2 that have been FDA approved, fully FDA approved so far in this country, lecanemab and donanemab, and there will be others to follow. And the thesis that we wrote about was the notion that it is no longer scientifically sensible to think that the amyloid is a byproduct or a secondary traveler or something that is not crucial to deal with medically in this disease. And in the article, we listed 13 peer-reviewed findings that I think have virtually no controversy behind any one of them, that amyloid buildup occurs very early in the process that we define as Alzheimer's disease. Indeed, amyloid accumulation, like tau accumulation in tangles, is part of the definition, or a principal part of the definition of Alzheimer's, as it's now been increasingly refined and revised. And so the debate over time about whether amyloid lowering can work in this article is put squarely in front of the reader as something that is very well supported scientifically and is analogous in some ways to other elements of chronic disease that regulatory agencies have recognized as important biomarkers. The example we cite, which is an obvious one, is lowering LDL cholesterol and dealing with atherosclerotic disease with HMG-CoA reductase inhibitors, which were first approved around 1985, 86, based on the fact that they dealt with this very key biomarker. You could call it a surrogate marker, but I would say that LDL cholesterol alteration, dyshomeostasis, is a fundamental etiological factor in the disease. We know that at that time and subsequently, people rightly argued that atherosclerosis is a very complex disease with inflammatory components, with altered calcium homeostasis, with a whole range of cytokine and acute phase protein alterations, not just a matter of dyshomeostasis of lipid lipoprotein metabolism. And nonetheless, the regulatory agencies around the world steadily approved HMG-CoA reductase inhibitors that have now obviously become of use to many, many millions of people. Now, we've been told in the Alzheimer field for decades that clearly we're not there in Alzheimer's, and we do not yet have widespread agreement, not only within our field, but importantly also by people who are knowledgeable about biomedicine outside of the Alzheimer field, that we have a surrogate marker that could be likened in a sense to LDL cholesterol lowering and be the basis for approval as a surrogate marker or as a etiological event. And this article attempts to lay out the argument as to why we are very close now to accepting the notion within our field, but also outside of our field in medicine, in the pharmaceutical industry, and I think increasingly in regulatory communities, that amyloid reduction is a fundamental way to attempt to slow the progression of Alzheimer's and, importantly, potentially to prevent its symptomatic advance in secondary prevention, so-called, and ultimately to prevent its deposition in the brain in the first place in a sense of primary prevention. So, George, I would say that the argument tackles a topic that's been fiercely debated for 4 decades, whether amyloid is causative, whether it is an epiphenomenon or a secondary or tertiary event, and depending on if it is causative, whether it is therefore worthy of aggressive medical assault. attempting to remove it. And we opine in this article and give reasons for our view that amyloid reduction, as we see now being driven forward in prevention trials for Alzheimer's, which is terribly important, and one of the missions that you mentioned, George, that this group has, that these amyloid reduction attempts to prevent symptom onset in what is sometimes referred to as secondary prevention mode. There's different definitions of that is going forward. And we're very excited about that. At CTAD, just a week or so ago, we heard the full enrollment of one of the major trials, the AHEAD 345 trial in 79 centers around the world. And so we very much believe that both for symptomatic Alzheimer's disease and for pre-symptomatic Alzheimer's, we are in a good position if we attempt to lower amyloid, show that it's lowered, measure other elements of the disease, such as tau. We have this really very useful plasma biomarker and p-tau217 ratios. Measure GFAP, glial fibrillary protein, which also can respond to amyloid lowering, and then measure cognition if patients are already in the clinical phase of the disease, or pre-symptomatically make our judgments based on the degree of amyloid lowering coupled with these other biomarkers of an amyloid-driven process. So I'll summarize my remarks right now. Look forward to questions and discussion and other points of view, to say that we, as a group of 12 authors, each of whom has worked on this disease for years and decades, feel that amyloid lowering has arrived now as a biologically and medically validated surrogate marker for interfering with the progression of Alzheimer's writ large, for the Alzheimer biological process. And we know that this needs to be debated, and this article is an attempt to bring this debate even more sharply into public focus and hear the pros and cons of making amyloid lowering itself the key surrogate marker that could ultimately be similar to some cancer agents, some cholesterol lowering agents, some infectious disease agents, where you don't wait until the symptoms are starting or certainly full-blown to deal with the biology.
So Dennis, I'll start with one question, but obviously we'll open this up very much to hopefully a real vigorous discussion. Not everybody who has amyloid in their brain ends up with symptomatic Alzheimer's. So if you're going to basically target pre-symptomatically amyloid, will you not be treating a number of people who may not, in the end, have gotten Alzheimer's symptoms? And how does that compare to other treatment areas in which the same problem probably occurs?
Right. So the same problem does occur with other surrogate markers that have been used for approval for general use in the public. So obviously, a great many people who take statins, and statins are not without some side effects, especially in some individuals. Many of those individuals will not necessarily, and would not necessarily, even without a statin, have succumbed to severe atherosclerotic disease, heart attack, stroke, peripheral arterial disease, etc. And the point about amyloid and Alzheimer's is that indeed, there are people who carry around amyloid loads in their brain. Many in the field loosely call this a head full of amyloid, without yet, or maybe ever, having a substantial deficit in cognitive function. But many of them do go on to cognitive dysfunction. And of course, with people living so long nowadays, commonly into their 80s and 90s and even beyond, a lot of people who have amyloid buildup at 50 or 55 or 60 or 65 will likely develop some telltale symptoms of cognitive and memory impairment. We're not advocating in this article that we're ready for general primary prevention with an amyloid-lowering agent in all people in society. But we're saying that the current paradigm for carefully quantifying amyloid load in individuals and then seeing whether a therapeutic agent can lower that load even prior to symptoms, that should be an outcome measure that will be followed closely and hopefully acknowledged by regulators. And of course, that's the basis for the secondary prevention trials that are underway. The outcome of the AHEAD study, one of them will be the AHEAD 3 study will be amyloid lowering by PET scan will be the principal outcome measure, and then other outcome measures with it secondarily. And then in people who are a little bit more advanced in the amyloid in the AHEAD 4-5 study, it will be advent of cognitive symptoms and how you do cognitively. So I think the answer to your question, George, is that there are many examples of where we treat sometimes with agents that can have significant side effects. We treat people pre-symptomatically to avoid the development of disease. And certainly some of the feared infectious diseases, obviously we deal with those with vaccines, which is where, in my opinion, and I think many people on the call will agree, we want to go with Alzheimer's as well, if there were a way to safely prevent the onset of clinical symptoms. So it is true, not everyone with amyloid will develop full-blown Alzheimer's disease. Many will. It's one of the very strongest risk factors. If you couple that with ApoE4 as a genetic predisposition, you have two factors we measure every day when we treat patients in our clinics, as I and my colleagues do. We measure their ApoE genotype, and we measure their amyloid load with amyloid PET or with spinal fluid analysis, and then we initiate amyloid treatment. Now, this is in the symptomatic phase. That was highly debatable until a couple of years ago. Now it's increasingly accepted that this is a way to deal with Alzheimer's. I think this movement towards secondary prevention, if we call it that, and ultimately maybe to primary prevention if the agent is safe enough, is something that the Alzheimer's field should very much incorporate.
Martin Landry has a question in chat, which I will pose. Martin, as everyone knows, was deeply involved in the recovery trial in the UK in preventing COVID or at least serious COVID symptoms. So Martin asks, can you comment on he compliments your presentation, but oh, wait a minute, just a second. It moved out of sight range. You say amyloid should be a surrogate, but what you haven't commented on is where you find the amyloid. Is it in the brain, CSF, blood? Could you just expand on how you would identify the presence and the presence of sufficient amyloid to deem it an adequate target for a surrogacy?
Great question. I think the current best and most concrete way to do this is with amyloid positron emission tomography, amyloid PET scan, where you actually see the level of amyloid. Just this morning in my medical center here at Mass General Brigham, we have plaque and tangle board each Wednesday morning, and we look at amyloid PET scans and MRIs to decide if borderline patients qualify for going on Leqembi. And now we just today actually got approval in our medical system of starting patients on donanemab. So amyloid PET scan is a well-validated indicator that you have a certain level of amyloid, and an amyloid level that's roughly higher than 25 or 30 centiloids, or maybe even a little less than that. Many of us in the field believe that even lower levels, centiloid levels of amyloid, will be problematic. But at a certain level, we will see a patient who we have to worry about will advance. But we don't have to rely only on amyloid PET. And of course, amyloid PET is a very expensive and difficult diagnostic and monitoring approach for this very common disease. It's fantastic that through a lot of biomarker research, we've identified phospho-tau 217 in plasma, often expressed as a ratio to total tau 217 at that epitope. That elevated phospho-tau 217 ratio is a strong indicator that a person is developing high levels of amyloid in a way that deserves treatment. And we know that the prevention trials have used this as a way to decrease the number of amyloid PET scans they need to do to enroll people in prevention trials. I think there will be other markers as well, and hopefully more and more they will be in plasma. We have very good CSF biomarkers, namely measuring A beta 42, tau and phospho-tau in the CSF, which we've done for a couple of decades now. And they're, if anything, even more sensitive to amyloid buildup than amyloid PET is. But I would say that plasma is the way we have to go, and we are going. And I think that PT217, I also think that glial fibrillary acidic protein, or GFAP, is another marker that is sensitive to amyloid buildup. And there will be a number of others. There already are some. So physicians can offer patients plasma tests to ask the question, are you on a trajectory that suggests that your amyloid deposition is going to be problematic later on? And of course, this can also be done for non-amyloid driven diseases, other brain diseases, other tauopathies, for example, can be also monitored by plasma tests in an early stage in clinical evolution. So it's not just about amyloid, it's about this serving as an example of other neurodegenerative diseases where other biomarkers, hopefully measurable in plasma, will be useful. So I think that's the answer. We don't want to rely solely on amyloid PET. We want to rely on plasma biomarkers, and those have yet to be fully approved by the FDA and other regulators, but I think they will be, and then they will come into common use because we can't do amyloid PET on any more than a fraction of people who are enrolled in carefully organized prevention trials.
This is a follow-on question from Diego Silva related to this. Diego is from Vaccine, the Australian biotech located in Adelaide, producing vaccines and immunotherapies. He asks, can you comment on the magnitude of effect of a currently approved therapies and whether amyloid blood quantification is a useful tool for monitoring the small effects seen so far? It's sort of the question of how much amyloid do you need in order to have the efficacious removal proved to be a surrogate marker, and how do you quantify that with non-amyloid PET technologies of measuring amyloid load?
Well, it turns out that, again, phospho-tau 217 levels in plasma correlate quite nicely with the degree of amyloid buildup. And so that is a peripheral marker. There'll be other tau isoforms that will be useful. I mentioned GFAP. So I think there's a way of quantifying how bad is the amyloid load short of getting an amyloid PET scan, which is the current gold standard. All of these plasma and CSF biomarkers are compared to the gold standard of amyloid PET. And so we obviously need to move the field into more and more utilization of sensitive plasma markers. And there'll be other new ones coming along, and there'll probably be a panel of plasma biomarkers. In terms of the effect size, which I think the question raised, how useful is amyloid lowering in people with Alzheimer's? And that is a moving target. We know that the numbers for the approval of lecanemab, for example, in Clarity AD was about a 27% slowing of worsening on CDR sum of boxes. But importantly, activities of daily living in that scale that was used in that trial was close to 40% slowing. And then we have the recent evidence that Chris Van Dyke of Yale presented at the AAIC meeting in August, that some patients in the Clarity AD lecanemab pivotal trial, phase 3 trial, that had particularly low levels of temporal lobe tau measured by PET scan, or had relatively lower levels of amyloid on PET, still abnormal, but below 60 centiloids, those patients, when followed for 24 to 36 months, did quite well. That is, many of those patients, and they were talking about numbers in the hundreds, 300 or so, in this Clarity AD study, who were mild at entry, had virtually no worsening over 24 to 36 months. And that's very encouraging. So when the effect size from the original Clarity AD trial is quoted as a 27% slowing, obviously my patients and anyone would be a bit disappointed. We want to hear there's a 40% slowing or a 50% slowing. And in those people we treat very early who have mild disease, but do have clinical Alzheimer's, bona fide Alzheimer's already, symptomatic, if they're mild, including their low levels of tau spread and their relatively lower levels of amyloid buildup, they do particularly well. There were even some patients in that category that got a benefit or improvement, something we thought we'd never see in amyloid lowering. So I think the effect size is modest in many patients right now, but only through our clinical experience today in rolling out these agents and seeing how patients do over the first year or two, will we be able to say, what was the degree of slowing that we achieved? And I think in some patients who are very mild, the slowing is great, and that augurs well for the prevention trials, Trailblazer 3 that Lilly is running, and HEAD 345, which is an academically based consortium. I think they're going to do quite well. And frankly, I don't want, part of the reason of this article, I think the authors felt we don't want to wait until the results of these prevention trials come out, and then people say, my goodness, you mean if I have such and such a level of amyloid, I'm really at the risk of going forward. And if I lower that level, I'm not going to develop near as many symptoms or much less symptoms. We're going to hear that in roughly 3.5 years or so. There may be peaks before that on the data. And I think we want to have this argument resonate with the informed public and with scientists and regulators soon, that amyloid lowering is likely to potentially slow down or prevent the beginning of clinical symptoms when you're still asymptomatic.
And a related question coming from Cynthia Smith, but what about the side effects? The side effects can be serious.
Right.
So we're going to introduce an amyloid lowering agent either in the early symptomatic stage or pre-symptomatic. How do we deal with, at the moment, what appear to be some significant side effects?
Right. So that's a big part of this thesis that we're trying to advocate for using amyloid as a surrogate to say a patient should be on treatment and should see how they do on it. What are their risks? And with amyloid lowering antibodies, virtually all of them tested so far, some waiting in the wings, there has been ARIA, amyloid-related imaging abnormality, edema or hemorrhage, or both. And this is a significant side effect that we have to try to keep under control. And the way that the EMA, for example, dealt with that was to exclude patients from treatment in Europe, we believe, based on their decision, who are ApoE4 homozygotes and therefore have a higher risk of ARIA. We treat them in the clinic that I work in. We treat E4 homozygotes just as we treat E4 heterozygotes and non-ApoE4 carriers. And we watch them very closely. And the early data from CTAD that just was announced was that the risk of both radiologic ARIA and symptomatic ARIA was pretty much right along the lines of what Clarity AD showed in its 1,800-person trial. It was not worse. We worried that when you took an agent like lecanemab into the general population, things were less regulated, less controlled in our trial. There was more comorbidities present. But so far, with careful administration, we don't have an excess of adverse events of ARIA. Now, we've all heard about patients who've died. Some of those patients died because the ARIA was not recognized. They were also anticoagulated or given TPA or another clot buster. We all now know that that has to be avoided. All of our patients carry a card with them. If they go to the doctor or to the emergency room for any reason, they right away announce that they're on an amyloid-lowering drug. And we're trying very hard to cut down the bad events. ARIA radiologically, in the case of lecanemab, we have the most experience with, is asymptomatic in about 3/4 of cases. That is, the general frequency of ARIA-E is 13% in the Clarity AD trial, and only 3% of those patients had symptoms. And most of those symptoms were not terrible. They were headache, dizziness, confusion, things you don't want to have. But when you see them, you right away ask for another MRI scan, which is routinely available widely. And if the patient has ARIA coupled with symptoms, certainly in our center and in most centers, and the appropriate use recommendations suggest this or indicate this, we pause the medicine. And this morning we made 2 or 3 decisions where we paused treatment for at least a month or 2 until the ARIA clears. So to summarize, there are side effects of treating. You especially don't like to have side effects in people who are currently asymptomatic, which is what happens in so-called secondary prevention mode. The people are not symptomatic from their Alzheimer's, and we really feel nervous about inducing symptoms of ARIA. But it is happening in those prevention trials. We are following the patients closely. We can stop dosing. We can stop it permanently, or we can pause it for a month or 2. The symptoms become obvious, and frankly, the asymptomatic ARIA that can occur, we also watch for that by getting routine MRIs before the 5th, before the 7th, before the 14th, and with donanemab, it's going to be more frequent at the beginning of treatment. So there's a method to watch out for adverse events, and I'm very hopeful that subcutaneous administration is not far away. We know some agents already are trying to qualify for subcutaneous administration, and that might also be associated with less morbidity to patients. So I think side effects are problematic, but can be monitored and controlled with lots of good medical education and close monitoring.
Martin Landry, you have a long comment here on an earlier answer that Dennis gave pertaining to if you go earlier and you find you can reduce amyloid by X, you get a potentially bigger throughput. Would you care to go off mute and just make your comment? Okay. He basically says that the same thing that you were commenting on here may have held true with LDL. The more we lower LDL, the better effect we get, right? And so he's making an analogous comment supportive of yours that by greater reductions, you'll get a better clinical benefit.
Yes. I can comment on that in saying that in the trials of the current agents, both beginning with aducanumab and then lecanemab and now donanemab, that there has been an attempt to lower amyloid as much as possible, getting below 20 to 30 centiloids or so. That is associated with better clinical benefit. One has to achieve that. One of the things that has always been a issue and problem in my experience has been the notion that you've done lots of amyloid trials in this field and you've struck out time and time again. You don't do it. And the answer to that is it's not correct. In the vast majority of trials that were done in the last 15, 20 years, either amyloid imaging wasn't available, it wasn't used, it wasn't a real indicator of how you did. And so a lot of the trials that failed that were putatively anti-amyloid could not be said to be anti-amyloid because there wasn't any direct evidence for that. Now that we have PET scanning, to tell us what's going on, you need to get down low, absolutely, like the example that Martin mentioned of LDL cholesterol. And so I think that, it's like preventing infectious diseases by having adequate immune titers. So if people don't get good responses to their COVID vaccines, if they don't get a change in their CD4 positive T cells when they get HIV therapy, they're not going to get adequate prevention and protection. So amyloid lowering, and probably a surrogate of amyloid lowering, will be phospho-tau 217 in the plasma that we are starting to measure in all of our patients who've been on treatment for at least 6 months in our clinic. How are things going? Is it going lower? And we want to drive markers of the amyloid process as low as we can.
There are a couple of other co-authors that are on this call. I'd be interested in hearing their views. Jeff Cummings, I know, is on the call, or at least was on the call. I just would ask if he would be interested in making a comment as a co-author of this paper or on anything that Dennis has mentioned.
Absolutely. And thank you, Dennis, for both the paper and the discussion. It is the best synthesis of amyloid data in the literature, so it's terrific. What I wanted to ask, this follows up on the earlier question about treating patients who may never become symptomatic. And there is some indication that people who have higher tau levels are the ones who are going to become symptomatic soon. And you would have greater confidence that those patients might benefit from treatment. So do you see a second biomarker like tau coming in to help us make decisions? Or, how do you see that evolving body of literature?
Yeah, Jeff, that's a very important issue. And I think the answer is yes. We don't want to rely just on amyloid estimation, but we want to look at the downstream effects of amyloid. So I think nowadays we can say from a biological point of view that there are a number of downstream effects of the initial buildup of amyloid, and the biomarker profiling that has been done in Cliff Jack's work and in the Diane study, et cetera, suggests that you want to watch what comes next after amyloid buildup. And what comes next includes changes in phospho-tau. And I think our paper already states that we're not just talking about amyloid as a lone surrogate marker of an indication for approval of an agent coupled with safety, but more than that, a change in tau, and it could be tau PET. I'm loath to recommend both amyloid PET and tau PET as something we would do widely because it's just not economically and practically feasible. But I absolutely think that measuring tau, we have some good ways to measure it now, right alongside amyloid, and measuring A-beta 42 and 4. There's evidence from some of the clinical trials that A-beta 42 levels, which are low in spinal fluid, for example, go up as you successfully remove amyloid with an amyloid-lowering antibody. So 42 levels rise, and that can happen in the plasma as well. So we will have a suite of fluid biomarkers accompanying these amyloid treatments, amyloid-lowering treatments, that will tell us, is this a good patient to use it for? And I absolutely agree. You want to be cautious. This is not an advocacy of broad, widespread, general treatment of everyone who's over 60 with an amyloid-lowering agent. That's not what we're talking about. We're talking about carefully selecting patients based on their amyloid load, based on the markers of amyloid trouble, like tau, GFAP, and selecting patients carefully, not doing it in any kind of wholesale fashion. We all love the idea in the future that we would have a vaccine that's safe and efficacious and could be widely administered to people with family histories or ApoE4 alleles, but we know that we're not there yet.
Thank you.
Speaking of these familial trials, I see that Randy Bateman is on. Randy, do you have... if you come off, do you have any comments or thoughts or questions of Dennis and the paper that you co-authored? Okay, absent that, I'm going to ask the question. This is a lawyer speaking, a patient advocate speaking, Dennis. So take this for the ignorance of the person who's going to ask it. There must be something happening in the brain before cognitive symptoms occur. That is, there must be some signaling somewhere in the synapses or at some molecular level that cognitive impairment will be soon coming.
Mhm.
Is there any research to your knowledge about sort of this 2 to 3 years before cognitive symptoms arise, being able to identify a marker that says symptoms are coming in 3 years, symptoms are coming in 2 years? Is there anything there?
There is, George, and actually it's a very thoughtful question. There are both biomarkers and particularly sensitive tests of memory function and cognition, even behavior, that indicate that a person is on a bad path. And they're still asymptomatic. They're still working, for example. They're still functioning in everyday life, but they have telltale signs that they're going to get into trouble. One of those, again, that we've mentioned several times, is phospho-tau217 elevation, because that seems to be an indicator that their amyloid process is not going well. It's not static, but it's dynamic in a bad way and causing more trouble. There also are subtle cognitive alterations that happen very early on. And that's what's being watched for in the AHEAD 4-5 study, where people already have substantial amyloid levels. We're watching to see how they progress in terms of subtle cognitive changes. And think all of those kinds of things, really phenotyping patients much more carefully and in a nuanced way, not offering everyone in society an amyloid-lowering agent. That's not about to happen, and I wouldn't want that to happen personally, but rather refining the people who are on the cusp of getting worse, who are most vulnerable. As I said earlier, maybe eventually we will have something safe enough that we can use it in a very early pre-symptomatic disease and even preventing amyloid. But right now, combining very sensitive cognitive measures, word retrieval and a number of others, as well as the patient's subjective feeling. We know now that people who feel like they're just not as sharp as they were, we have to respect that. And coupling it with changes in tau, GFAP, et cetera, we'll get an indicator of who in particular deserves to get an amyloid-lowering agent.
Great. Here comes a question on a completely different technology. Do we know if people on semaglutide for diabetes have a lower amyloid PET signal than those who are not? And could that be a signal about their risk for AD, or maybe even semaglutide as a potential agent that would be a prevention technology?
So there's a lot of discussion of that now in our field and in the metabolic disease field, of course. And the jury is out. We don't know yet. I think the most accrual of evidence so far has been that Ozempic and agents like it may have an effect on tau alteration, phospho-tau, et cetera. And that's a key feature of the amyloid cascade, basically. So I don't know whether amyloid levels, there are some mouse studies in which amyloid levels were influenced positively by agents like that. But I think it's more likely that other features of the downstream pathology, maybe tau, maybe inflammatory processes. There's a reasonable likelihood that these kinds of agents will help inflammation in the brain, as they do in peripheral tissues, oxidative metabolism, more than the primary pathology. So I don't think they'll directly compete, so to speak, with amyloid-lowering antibodies or with a gamma secretase modulator, which I think is a very important therapeutic approach. But they may help secondary and tertiary events that come downstream from that. But we don't know yet, and we'll certainly be able to find out because these agents are now incredibly widely prescribed, and we'll be seeing a lot of people who carry 2E4 alleles or 1E4 allele or have a family history of Alzheimer's. We'll see how they do, whether they have an apparent delay in their advent of disease.
Yeah, and we know Novo Nordisk now has two very, very large phase 3 trials in testing semaglutide as an agent to reduce or affect progression in Alzheimer's. So we will see what the outcome of those trials are at the end of next year. So I think this has really been a really good discussion. We just got a new comment. Has the use of steroids for symptomatic RAE been discussed?
It is discussed. Happily, in our experience here, we've treated maybe close to a couple of hundred patients now. We've done a couple thousand infusions. We've not come to a case where we needed to, other than maybe one case I can think of, where we needed to give steroids to suppress the negative effects of RAE. So this would be when you had radiologically moderate or severe RAE on the MRI, and it was accompanied by bad symptoms. Then we would certainly consider a course of methylprednisolone IV, followed by oral tapering prednisone or steroids. And absolutely that is a part of the management if there's a rather severe case of ARIA. Happily, the vast majority of cases have not required that. I know Larry Honig has reported his results at Columbia in their practice, and they've had very few or no serious cases that required emergency room visits, ICU, hospitalization, etc. But yes, the answer is we have appropriate use recommendations that Jeff led for lecanemab that mentioned the use of parenteral steroids followed by oral tapering courses to control bad cases of ARIA. We're trying hard to avoid those. George, if I might, I'm going to close in a moment. I wanted to make the point that I think that this article is not only about advocating for using amyloid lowering as a surrogate marker for ultimate approval of Alzheimer's, particularly for agents that have a similar mechanism of action to the 3 that have already been approved by the FDA. That was one of our main theses. But part of this exercise and this discussion today is to get the world as a whole more comfortable with the notion that amyloid is almost certainly an etiological factor in the disease, a driver of the disease. And we want the public to understand that. We want medical doctors to understand that, because there's been so much, if you wish, fake news and incorrect information that amyloid is just a secondary or tertiary or fellow traveler. So I'm thinking of not only the issue of regulatory approval of agents based on thoughtful surrogate marker utilization, but also the notion that lowering amyloid, and we write this in the article, should be thought of front and center when we look at trial results, because lowering amyloid is taking away a causative agent of the disease. It isn't the only biological driver, but it is a very important one. And at this point in our knowledge, it's the earliest one that we can see drive disease. So we're trying to socialize the notion that amyloid lowering per se is a worthy goal, regardless of exactly how we do it or how we measure it. And we need to make sure that people no longer say, we don't even know if taking the amyloid away matters. We know people walking around with a head full of amyloid and they're okay. Amyloid trials failed right and left in the last 2 decades. All of those points have been dealt with in part in this article and in other literature, and they're not on the mark. They're really off the mark. So like amyloid in the heart, transthyretin, my colleagues who worked on that over the years said there was much less debate, less Sturm und Drang around the notion that amyloid in the heart is a bad thing and you've got to get rid of it. Everyone agreed, and it's now worked. There are 3 FDA-approved agents that deal with transthyretin amyloid. So that was one of the goals that I had certainly in this paper, was to make people more comfortable with the notion that amyloid is a driver of the disease. It can be viewed as a cause, and it should not be accepted as anything that we can let go and not do something about.
Well, I think you've done a masterful job in this paper of both making that claim. I say it's a claim, but that demonstrating that proposition, as well as trying to respond to those who would suggest something to the contrary. And I know that your goal in getting this out there is it is a goal of this patient advocate here that we get this discussion going now, because we don't want to wait for the year or two that we're going to have to wait after a successful prevention trial. So I think I may have gotten cut off, but if I haven't, let me just thank everyone for attending.
You're still there. You're still there, George.
I just have the different screen now. So this paper is now live. There is a QR code here for its access. I think we've also put a link to it in the chat. And we very much welcome comments either in private back to Dennis and the co-authors, or in public to get this discussion going, to make sure we have a thorough discussion of this, and hopefully come to the view that we can use amyloid lowering as a surrogate for final approval or traditional approval at the regulatory agencies. It is still a controversial matter, at least at the regulatory level, but less so at the scientific level. And so we hope that people will attend to this. We will be taking other steps to make sure this paper is widely circulated and that it generates the kind of discussion that we need to have in the scientific field, which will give regulators the basis, we hope, in time as these earlier interventions pre-symptomatically or pre-pathology come through the trials that are currently ongoing in pre-symptomatology, to give them confidence that they can act in this fashion. It is only one, but a major one in getting Alzheimer's prevention into the patient community. So thank you all for attending today. Deeply appreciate your attendance here and for your active contributions to this discussion as we go forward. Thank you, Dennis, for your work. Really deeply appreciated.
Thank you.
Bye-bye all.
Good to be there. Bye now.